Welcome once again to IndieBio's Fundamental Questions of Biotech series. Thank you everyone for coming and attending today's event, which I think will be extraordinarily interesting. My name is Julie Wolf and I'm here with the new program that IndieBio has built out in New York, but also here virtually in this little corner of the internet where we like to host events. So before I introduce today's panel, I'd like to introduce you to the event space that you're visiting, just in case this is your first time spending time with us today. As you scroll down, please notice that the bios of today's panelists are there, as well as different links related to the companies and the institutions where they work. So we've done a little bit of the legwork Googling for you and adding the links directly down below. As you scroll down, you'll see that there are two additional buttons, one of which is an invitation for you to sign up for IndieBio's event newsletter. So should you want to attend more events such as this one, we send that information directly to your inbox once a week, just with the events where you can participate with our community. You'll also see an application for IndieBio's second cohort. I think it just says application process because, of course, we in the New York side are working on our second cohort, and in the San Francisco side, they are looking for their 11th cohort. And we are both actively recruiting right now. So if you would like to become a part of one of the cohort companies, we'd be happy to have a discussion with you, and you can learn more by clicking that application button. Let me also say we are about to present our first cohort on the New York side and the 10th cohort, believe it or not, from the San Francisco program next week. So if you'd like to learn more about how you can attend the Demo Days event, it's actually a 2-day event, go ahead and click on that IndieBio newsletter and we will send that directly to your inbox, or go to indiebio.co/events. All right, enough promotion for IndieBio. I think we're all here to talk about the very interesting question of where our future drugs, where our next drugs will come from. We've got we've got two thought leaders who are CEOs and one the director of an institution who are going to address this from their various angles. So I would first like to say thank you to our panelists and welcome. So to my left, although I'm not sure how you're seeing it, I see Nicole McKnight, CEO of Stellate Therapeutics. Nicole, thank you so much for taking the time to speak with us today.
Yeah, this is exciting. Thanks to IndieBio for coming on the scene in New York City and really helping to grow the ecosystem here, and really excited that, time goes by and you're on your second cohort already. That's really great. So, we think the more the merrier, and in New York, we're not competing against each other. And I've been a part of the growing biotech ecosystem in New York for going on 10 years, and it's drastically different now than it is. And we believe all together in the rising tide raises all ships. So, there's a lot of collaboration, a lot of support, just trying to bring more and more funding and resources into our ecosystem.
Yeah, absolutely. Very, very nicely said. And I agree. It's been a completely collaborative effort, as I've met people such as yourself who are part of the New York biotech ecosystem. Yeah. Also joining us today, Damien Kudella, CEO of Cayuga Biotech. And I should give a disclaimer here because Damien is part of our first cohort of the IndieBio class from New York. Thank you very much, Damien, for joining us today.
Happy to be here, and hopefully the only ever IndieBio cohort that doesn't actually end up in New York.
Oh, that is a good point. We've all been living a long time through these Zoom calls. Hopefully today's will be very interesting for everyone to listen to, but we know that Zoom fatigue is a real thing. And finally, last but not least, I'd like to welcome Pete Meinke, the Sanders Director of the Tri-Institutional Therapeutics Discovery Institute, or TDI, located here in New York City. Pete, thank you so much for taking time to be with us today.
Sure. No, it's a pleasure to be here, and I couldn't echo your comments more. Of a rising tide lifts all boats. I actually joined TDI because I've been offended for years that New York doesn't have a thriving biotech sector like other parts of America that shall not be named in this call.
Shall not be named. I like that. Great. So I usually like to begin these conversations with adding a little bit more detail to your own scientific background and your experience within biotech. And so I rather than me reading something off, nobody knows your own bios like you guys do. So I'm going to hand it right back to you, Pete, in order to talk about your experience and some of the reasons why you may be offended that New York hasn't had a biotech ecosystem and what you're doing at TDI to help increase that ecosystem footprint.
Sure. No, thank you. Briefly, I'm a chemist by training, and that included a postdoctoral fellowship at Columbia, because I thought I'd be an academic. But I was lured into big pharma, Merck, where I spent 28 years. And I worked in most therapeutic areas during this time, and my team put 13 compounds into a clinic and one drug, elbasvir, for hepatitis C. But as I said, New York never had a thriving biotech system. And I saw this as a huge crisis as our young people were leaving the state when they finished their training. This is profoundly negatively impactful. So in the latter part of my time at Merck, I was responsible for all of Merck's external medchem, which was over 500 people. And after 6 years of commuting to China and India, I moved to China for 4 years. And while there, I learned about TDI. And so I resigned/retired from Merck, joined TDI 4 years ago and became its CEO. We're a not-for-profit. We are owned by Rockefeller, Memorial Sloan Kettering, and Weill Cornell. We are funded substantially by philanthropy and our parent institutions, but also our industrial partner, Takeda. We're modality and therapeutic area agnostic, which means that while most of my programs are oncology focused, I can work on rare and neglected diseases as well. And we do many of these sorts of things. But here's what we bring to the table. I have a 40-man team split equally between biologics and small molecules. We bring industrial rigor to the academic community because they have this incredible insights into disease. They spend years, if not decades of their life studying a pathway, but they typically lack the resources and the experience to translate these ideas into animal proof of concept and therapeutics in a way that actually would make them able to license assets or create new codes. And so by being embedded in the community, we bring this expertise. I do not need to be a master of a disease area as these faculty are. Despite my experience, I rarely can keep up with them, but we actually work at the interface, and that's where the secret sauce is. Pharma lacks this type of deep biological expertise. And so TDI has helped create 2 new coasts, Quintus and Sperian Biosciences. Quintus is already closed, unfortunately, but Sperian is still in existence in New York. We've licensed 7 assets, small molecule and biologics, to large pharma and biotech. And we have 4 molecules, 3 biologics, 1 small molecule, deep in licensing negotiations right now. And I was told today a term sheet was tendered for 2 of them recently.
Oh, congratulations.
This is what we do. And our goal is to be a feeder pool for New York City biotech. So this to me is a virtuous thing.
That is really interesting. And I think that your experience both on the pharma side as well as pharma end of the table, as well as the experience you have now at TDI will add a lot to today's discussion. And I'm guessing that that might resonate a little bit with Damien, who I think has some of that deep academic expertise and is now bringing that to the clinic using, IndieBio, I think, as the mechanism to help learn a little bit about that entrepreneurial experience. But Damien, why don't you give a little bit about your background and what you're doing with Cayuga?
Sure. So Cayuga actually came out of my PhD thesis. So I was at UC Santa Barbara. My specific area is materials chemistry. So I'm a nanoparticle guy and probably somewhat of a black sheep for a therapeutics panel. But specifically what we ended up doing, we were studying how we could use nanoparticles to improve our treatment of bleeding episodes. And so at the end of the thesis, we ended up patenting our lead compound, which we call Chi-1, and then moving it out to get investors. So I will say with Peter, coming from Santa Barbara, New York looks massive. So we've gotten a lot better, but I do echo some of his concerns that there has been a bit of a brain drain. So I'm from Bridgewater, New Jersey, and the old Sanofi R&D site was two minutes away. And now, they're up in Cambridge. So there's a lot now, I think, in this area, a lot of knowledge that can hopefully be better put to use now.
Very, very interesting. And finally, Nicole, if you could give a little bit of your background. I know that you've been involved in New York biotech for a long time, and how that road ended up with you joining Stellate.
Yeah, sure. So I grew up in Massachusetts, and I had the really great opportunity to join a startup in Cambridge, Massachusetts, after college. So I did 2 years as a lab technician at Beth Israel Deaconess Harvard Medical School, really making sure that I knew that I like to be in the lab and work with my hands. And so I joined this company. It was a R&D arm of a company called Gene Logic, which was based in Maryland. And we were doing drug repositioning way back. This was in 2004, before that was really quite trendy. And it was a bunch of Millennium pharmaceutical spin-off people, actually before they became Takeda, I think. And yeah, it was, I caught the startup bug. I really, really enjoyed working there. I was employee number 7, did everything from kind of building the tissue culture room to designing the lobby to, checking in packages to presenting at company-wide strategy meetings. And I knew to do what I wanted to do, I needed to do a PhD. I went to London and did my PhD at Cancer Research UK. If anybody's actually not at that stage yet, I highly recommend going to Europe for your PhD. There are set timelines, 4 years of work, and you cram a lot in, but at least the end is in sight. And then I wanted to move to New York for personal reasons. And I mean, I don't want to Boston bash, but I got kind of sick of it for 4 years, and so wanted a new opportunity. And so I joined a lab at Mount Sinai doing my postdoc, working on Parkinson's disease. And I studied autophagy for my PhD, which is a really interesting trafficking pathway that has implications in many different disease areas. And so I focused on neuroscience for that. And I was convinced that I was either going to start my own startup based on my work or join one. And it was very apparent very quickly in New York in 2011 that was not going to happen. So
and the startup scene has changed quite a bit, I'd say, even since 2011. Yeah.
Yeah. So you could get most of us into a room, a small room, to talk about these things. We used to talk about, well, what does New York need? And affordable wet lab space for startup companies seemed to be the thing that really was going to make a difference. And there was really no lab space besides at the academic institutions, and a lot of barriers for making that happen. So I took an entrepreneurial course at Mount Sinai there first, and teamed up with some other postdocs, and we formed a nonprofit to try and build a small incubator. We were looking in East Harlem, actually. We formed a 501c3, and I was being mentored by the co-founder of Lab Central, which is now the gold standard model for co-working and incubators in life sciences in Cambridge, Massachusetts. Mm-hmm. And so started working on that project. And then when my postdoc came to an end, I actually joined that group, the for-profit arm called Biolabs, and worked for 4 years to build what is now known as Biolabs at NYU Langone at 180 Varick Street in Hudson Square. So, that was everything from pounding the pavement, looking for 30,000 square feet of space that's manufacturing zoned in Manhattan and Queens. And recruiting our first companies, raised money from the state through the Regional Economic Development Group and Empire State Development, and then partnered with NYU Langone, raised money from the New York City Economic Development Corporation, so from the city itself, and then renovated 50,000 square feet of space over 2 floors in an old Soho shoe factory.
Yeah, it's quite an interesting space, the biolabs. There've been a number of events there that I've attended, and it's been very educational for people who are not even just members of that space.
Yeah. And yeah. So, you know,
how did you get to Stellate from biolabs, Nicole?
Yeah. So you know, in my journey, just trying to get back to a startup, really, I knew that's really what I wanted to do. I was introduced by a good friend to the CEO of Stellate. We used to be called Antibiotics. They brought me on as chief operating officer in May of last year to move this company, this French company, to the US, really start to push the development, the drug development, the striving towards the clinic and raising money in the US. And then this year, I took over from our CEO as CEO.
Yeah, oh, that's wonderful.
I was very, very lucky to find not only a startup that needed me at the right time at that very early stage, but also to be in neuroscience, you know, this field that I'm so passionate about. I mean, yeah, it's kind of slim pickings in New York still, although it's getting much better. But yeah, like I said, I was very, very lucky to find something at the right time, and also something in a great kind of field that I think is interesting. And just to give a little bit of a plug, I'll tell you what makes us different. Our molecules come from our own gut microbiome. So just to pull it back to you know, what brought us together here with these new sources of molecules.
Yeah, that's actually exactly where I was thinking to go. So this is a really great opportunity to segue to some of the actual discovery processes, which you have both Nicole and Damien participated in, and Peter have lots and lots of experience in terms of identifying some of these candidate molecules. And I think that maybe just piggybacking off of what you said, Nicole, the ability to, let's say, mine natural systems for new compounds is certainly an area that's very hot right now. But I wonder where the insight came from, either with your background in neuroscience or with your scientific founder's background, was to even look within the gut for something that would be a compound to treat diseases such as Alzheimer's and Parkinson's, which I believe is what your primary indications are for.
Yeah. So for us, I mean, it's the brainchild of our scientific founder, who has spent decades annotating genes from our gut bacteria and linking those metabolites and genes with biological function. So in France, you get kicked out of your academic appointment at 65. There's mandatory retirement. And Antoine Danchan, our founder, had lots to do. He's spent his life building this portfolio of molecules. And so then it actually gave him a clean break to start a company, raise a bit of money from foundations and get started, then really working on this portfolio. So it's over 100 molecules, metabolites that are the product of the bacteria in our own systems. And the lowest hanging fruit is our lead compound. And it's a very, very interesting small molecule. It's a modified nucleobase called QN. And it's produced exclusively only by the bacteria in our gut. Yet we have an enzyme coded by our own genome that helps it to do its function, which is being integrated into transfer RNA in order to really promote better protein folding. And so in neurons, you know, protein misfolding and the subsequent aggregation is especially detrimental in neurons, and especially as we age. So we believe that taking this molecule, we synthesize it chemically, and we're using it as a therapeutic at a higher therapeutic dose to really push this positive neuroprotective phenomenon to hopefully decrease protein aggregation and neuronal death.
Oh, very, very interesting. And just one more follow-up question before I pass the mic. Was this discovered in a screen for this function, or was it based on, let's say, the years of discovery and just kind of characterizing these genes one at a time? Since, I mean, the way that we study genes has certainly changed over the past decades.
Yeah, I mean, so we're completely switching the paradigm of drug discovery from like kind of the classic pharma model where you have a target and then you do drug screens, and Pete can tell us all about this. You know, you screen libraries, you know, millions of compounds to find some sort of modulation of this target. We actually start with the molecules themselves, and we have this 150 potential therapeutics, and then we do what we call indication screening. So coming at it from the opposite way, based on what little literature might be out there, some studies, or based on what it's doing, what it looks like, what sort of molecule it is, what can we guess, what indications, what diseases could these be best for? And then we do a lot of kind of in silico work, and then we start to do some in vitro proof of concept. Antoine still has his microbiology lab, so we can query what's happening, and then start us on the road to drug development and to the clinic.
Very, very interesting. Indication screening, that's kind of a new phraseology for me. So it's a very neat approach that you're using. I'd like to turn now to Damien and talk about your drug discovery journey that you've been on, in terms of not necessarily starting with a broad approach, like the gut microbiome is a very, very complex system, but so is the understanding of how different signal transduction pathways interact and all of the various ways that one permutation within those systems can have downstream effects. So can you talk a little bit about how your deep understanding of clotting and the clotting cascade and all of the different factors involved led to the discovery of your lead candidate?
Mm-hmm. So I think Nicole's on, I very much agree with her that, a lot of the hopeful answers are already existing in our body or somewhere in nature. It's just repurposing them to turn them into a therapeutic. But our bodies are what, millions of years old, and average person is 100 years. So when in doubt, trust the body, because they had much more experience. But specifically with coagulation, so we had my professor Galen Stuckey, again, he's a pure material science guy. He had actually not gotten into biology and clotting until about 2004. So typically with blood coagulation, you're looking at proteins, blood cells, trying to fix things, the balance when it gets out of whack. We kind of came at it from left field, and we happened to be working with another professor who was working with a polymer called polyphosphate and was having trouble, how do you deliver it? We knew how it was going to act, but getting it into the bloodstream, making sure that it ends up at the right site in the right dose was problematic. And so that was really my PhD thesis was we had a promising compound. It was then, how do we turn that from promising into something that we can actually put into effect?
And so then working in collaboration with folks, not only within your research lab, but going forward, you kind of were able to come up with those candidate compounds based on some of those natural structures.
Exactly, and frankly, the reason we ended up with Chi-1 being a polyphosphate-coated nanoparticle was because I originally tried all of those traditional proteins, tried to deliver them, failed miserably, cursed my PhD, and then one day our collaborator said, "Hey, why don't you try this polymer?" And it worked, and I was smart enough to just keep moving with it.
Yeah, once you find something that works, continue down that road toward drug development. Now, Pete, we have two stories here, one from an academic turned entrepreneur and one from a serial entrepreneur, very entrepreneurial spirit in Nicole. And I wonder how typical this story is for those that you work with at the TDI. either the identification process, but also some of the challenges in identifying which might be a good promising candidate.
Sure. No, they're actually very similar. But there's some very unusual distinctions, and I think actually I should probably explain them a little bit more. And so we had the good fortune, I'm Sanders director and CEO in honor of Lou Sanders, who gave a very generous gift that actually funded the initial formation of TDI, because he was actually on the boards of all three institutions. And he recognized that despite the extraordinary intellectual firepower that exists in the community, they had largely failed in commercializing their insights. They would publish top tier papers and there'd be limited translation to be translated by others. So he recognized that there was a cultural gap and an experience gap that existed in this community. So the initial goal of TDI was to bring in experienced professionals that actually have enough of the drug discovery experience that we can help guide our colleagues, our academic colleagues, so that they can actually create more rigorous assays, do more rigorous work. And I should actually point out that it's a really capital efficient model. And there's a point to be made here. So my staff, when we're a little understaffed, COVID's affected hiring, but we're normally about 40 people. But every biology team I work with, it could be as few as 3, it could be as many as 15. That is free to me. We, society, have paid for those, right? And so I get to leverage that expertise. And so what happens is when they're studying a disease process or they learn something, they find a molecule, they learn something about disease, because we've had everything happen, they struggle to advance those insights into an assay that is sufficient to advance the molecule to an animal model or de-risk it for safety and generate a data package that actually allows them to attract viable attention from the venture capital community. This is where our expertise came in. So when TDI was originally new, it was only 6 of us. I wasn't here at that time. And a lot of chemists, they would make molecules for the biologists. That was the idea. And then we recognized that their assays are not robust enough. They didn't understand the things that pharma takes for granted. We now have a hardcore biology group that actually helps them build the assays so that they are actually, the output of the assay is what they think it is. They make sure that the cell lines are appropriate as opposed to being borrowed from your neighbor down the hall, and they're not what you think that they are. And then we expanded to have a biologic swing. And just as a related element of this, 3 of my staff, myself included, are also Weill Cornell faculty, which allow us to write grants, be co-PIs on grants. This is really valuable for the community. So one thing we had with our biologics community is everybody that is interested in a biologics target, many of them are interested in CAR T. So just like making a bioassay, they thought, well, we can go and do it. And it turns out they actually can't, most of them, because the skills to go and do this are orthogonal to their expertise and they're not resourced. So we're faculty. We hired 3 postdocs, one of whom we had implement a CAR T platform. So now anybody that wants to actually go and look at a biologic, and we have the ability to make fully human biologics, either from our animal models or our phage library, we can also translate into a CAR T platform. So we try and augment the expertise of, like, for example, Nicole's founding partner and his decades of expertise. I can't replicate that. I can leverage it and bring the things to the table that are absent from theirs. And so we run 25 programs, which is really hard with a 40-man group. So you really have to focus on what is the key killer experiment to get to the next stage, or to send it, to stop the program, or send it back to the PI lab saying the biology is more complex. And this is what we try and do. And it's this constant balancing act. So it's a little bit different. But here's what's really unique is often when we work with the faculty, because they come out of a different background, they don't understand the logic of what we go and do. We have multiple repeat faculty because they see how the expertise we bring and the resources we bring augment their skill sets. And these programs can go really smoothly because all of a sudden it makes sense. And you start working in this seamless way where you're leveraging the strengths and capabilities of each other. So our starting points are not dissimilar to the ones that you're hearing about. They come from all sorts of sources. But programs that TDI will work on, we will have clinicians who are studying tumors, and they're doing the type of genetic validation that Nicole was talking about when you're looking at the GI cultures. And they'll say, we've made this intellectual leap. These people do not have the skill sets to make a primary assay or run a screen. This is what I spent my entire career doing, right? And you can start seeing how we adjust what we provide to augment a gap that exists in the community. And then the entire output is owned by the institution. So if they want to license something to a biotech or create a new co or license to pharma, we help support them because we write the technical packages, which anyone that's lived in pharma would be very familiar with, if that
Now,
helps.
is there a sweet spot, Pete? As you're talking, I can imagine that, let's say a researcher comes to you with a candidate molecule, but maybe they already have a little bit of in vivo data, right? Like, at what point would you begin the collaboration process with some of the folks about whom you're speaking?
All right, so we have the entire continuum. So we have literally on both small molecule side and biologic side, have people come in with, I'll use a small molecule example, but biologics exist, where scientists have tried to make a molecule against a tumor, and it failed, not for efficacy, but intolerable adverse effects. And so they spent years studying the biology, and they understood what everybody else had missed, that it led to the intolerable toxicities. So now you could argue they have advanced molecules that are, I'll say, non-selective for the purposes of this. And so we've got a starting point, but all the assays and everything else exists, but we have a biological insight that exists. But it is not uncommon that we will have someone comes in with a biological insight into a disease pathway that had previously been missed by others. And now we have to figure out a starting point. And so we'll do data mining, which for me is as much patents as the literature. So a lot of people don't do that, but that's what we do. In my group of 40 people, I have 3 full-time computational chemists with unrestricted access to Schrödinger software. And we've negotiated a license so that any scientist in the community, that means any scientist at the 3 institutions, can learn how to use it, and it's free to them. And we provide training several times a year.
I mean, that's worth hundreds of thousands of dollars, if not millions of dollars.
It's actually millions a seat for pharma. And so, but it's incredibly valuable because these scientists often allow me by saying, oh, Nicole's target is this. It is a family member, not terribly distantly related to this. And here are leads in the patent literature that people use to modulate, and we can go and dock it in here, and maybe you can get a toehold to start. And this type of thing happens all the time. And the same thing happens in biologics. It's really bringing pharma quality tools into a community of extraordinary scientists where they've never had that. And the other thing is, because I'm owned by the institutions, I can talk to any scientist, my staff can talk to any scientist, and there's no paperwork. If they choose to work with us because they see value, that's fine. If they don't, that's equally fine. There's never a wrong decision. And then we use a scientific advisory board to make sure that we get the insights that we need, because we have our own limitations. And it helps triage programs so that make sure that everybody agrees that these programs meet certain criteria to come in. But it forces us actually to be very, very focused on what we do. And because as a not-for-profit, while I'm well-resourced, so sit and think about 40-man team, at least 60 scientists that I employ elsewhere at CROs in one form or another, you can start realizing what the burn rate is. I have to be very focused on what we're doing. And I have on multiple occasions sent people home because they can't answer a question. And it's not because I'm angry with them. It's because they need to get away from my labs to get to where they can sit and think and focus on, why are we struggling? What is the problem that we can't solve? And so they can get to a place where they have time to sit and concentrate. And when I was the head of MedChem for TDI, I got sent home twice because I couldn't answer questions. And it wasn't because I was upset. It was because you have to carve out time to think deeply about problems to address them. You can't, multitasking in what I do doesn't really work. You end up doing a lot of things superficially to a superficial standard. And so we encourage people to really approach things in a different way. And we're really, really focused on solving key problems. Why can this compound not go into an animal, right? And then start, I don't expect people to solve the problem tomorrow. That would be naive. But I expect them to start formulating a strategy so we can get to a point where we can say, yes, we can solve this over time. Or, you know what, we don't have the skills, the resources, or the creativity to solve this problem and shut it down and focus on things that we can solve. And we do both. And it's a difficult conversation with faculty when we terminate a program. But it also drives us to do things I could never do in pharma, because pharma, and when I was there, and I hear this all the time from my colleagues elsewhere, will spend a small fortune arguing themselves out of an experiment. Whereas I can sit down with my teams and the faculty and design a time-bounded and cost-bounded set of studies that will get us an answer that allow us to go forward or not. And it's a godsend. Some of these will flame out. That's expected. And my board would be disappointed if that didn't happen some, because I wouldn't be pushing the boundaries. But it allows us to get successes that you would not otherwise get because you'd be too conservative. And so my group understands that they can fail and they don't have to worry that they've lost their job. That's expected. You have to go and do that. It's just what you need to do in our environment to thrive.
Yeah, that's very interesting. And I mean, a lot of what you said resonated with me, in particular, carving out that time for deep thinking that I think Nicole and Damien can probably appreciate is harder and harder to find in an interconnected world. So that's a very thoughtful approach to solving some of those very big problems.
And actually, if I could just interject one last little comment, and this is actually becoming a problem now with COVID. When I was at Merck, I'd be in meetings all day long. At the end of the week, I would get home, I'd be depleted, and I would look back and it's like, I didn't really accomplish anything. And so I was better at it now. We're actually struggling. This is why I've literally told people to check out at midweek Fridays when I can see the signs in my staff. It's like, you're not effective. Do something else and come back refreshed. You have to be able to do it. It's a mix of taking care of yourself, but also having time to actually really roll up your sleeves and try and address a problem.
Yes, recharging, 100%. Now, I would like to remind the audience that this is interactive and that there are questions. If you have any, please add them in. And there's actually a question here for you, Peter, about who the TDI works with. You've mentioned briefly that you work with the different institutions nearby, but there's a question here about whether you work with either, the question here is whether you work with non-New York based startups. I think there's also a question about whether you work with New York based startups. Do you work outside of that academic sphere? Or is TDI dedicated to academic research and moving academics into that startup world?
It's a great question. And I really should have actually added this in my introduction. Any project that I work on has to be unencumbered from companies. So I have to have, if we are successful, I have to have the ability for our institutions to license it in whatever way they want, which could include a new code. Any program I work on has to have a faculty sponsor. That said, many of the programs that I work on work with multiple New York faculty, US faculty, European faculty, because you might have the in vitro or basic biology experience here, but the animal model experience exists in Europe or California. That's fine, it's easily orchestrated, but there has to be a PI in my community who is the original founding one. And one thing that has actually not been an issue, but it just ties my hands. If a faculty creates a new co, I can't work on it. And the reason is as follows. Takeda pays a significant part of my bills because Takeda recognized, like lots of other large pharma, they cannot innovate to a vibrant future. So they're willing to pay literally half my bills for a seat at the table. And they're willing to pay for my tuberculosis programs and my malaria programs and my orphan disease programs they have no interest in, because it allows them line of sight for programs that are of interest. And when we get to the point where we've validated a program in an animal model, they are allowed a finite period of time to negotiate for that asset at market rates. And if they were able to get an asset every year or so, TDI is a bargain to them. But they have to satisfy what the institution and the faculty member believes is the right value. And we have optionality in the sense of, if a faculty decides the right path for me is a NUCO, that is retained. But guess what? That NUCO needs funding, and Takeda has watched it from its infancy. Guess who's going to be a primary investor? But because there has to be at least the potential for Takeda to license an asset that they care of, that's why my hands are tied in that regard. But if a faculty says, I probably create a NUCO, that's fine. They just can't have done it already.
They can't have done it already. No, that makes complete sense, given all the stakeholders that you're working with. Now, Peter, you mentioned computational biologists that you are lucky to employ at the TDI. And I think that one area I'd like to explore with our panel is where computational biology can speed along the discovery process, right? So not only the discovery, but even potentially part of the developmental process through in silico modeling or other types of screens that can now be done based on some of those databases that are available. And I wonder, I'm going to pass this over to you, Nicole, first, because I saw you nodding. So I think that you might have some thoughts on it, but also you mentioned that you have hundreds of candidate compounds. And how do you decide what to move forward with? I can't imagine that you are going to screen each one individually, in a test tube. Nobody really moves forward with drugs that way any longer. So do you use computational methods, or what other types of high throughput types of screening methodologies are you using to winnow down the candidates that you have?
Yeah, it's an interesting thing. We're right at this intersection right now. And it's this combination of really, really hands-on, super brain intensive work right now. But yeah, it started, Antoine used a lot of open source algorithms and ways to do his gene annotation, which led to this pretty rudimentary form of basically a Word document that has this 150 metabolites and, a paragraph or two around what it all boiled down to, all of those years and years of work. And then we've got a very, very intelligent team who is really mining that and going through and doing a lot of the hard work of looking at the publications. And when we're trying to narrow it down to 10 or 15 molecules to potentially outsource. So, it's really kind of a lo-fi right now. But that said, we are looking to partner with a company that can really kind of reverse engineer and validate what we've done. And so we have a great potential partner candidate for that. But for now, yeah, it's really kind of low throughput in silico, deep reading, and it takes a lot of brain power and a lot of kind of insight and experience from people. We had the idea that we would hire a bio informatician to kind of really streamline
some of those,
just kind of overhaul it, and it's a real asset for us. So we want to make it, but I think it's very naive to think that not only can you find a talented bioinformatician right now, not because they're so valuable, like that, you can't, yeah, if I would go back and do it again, or gosh, they are so in demand, and we need more and more of them to become really integral parts of our teams. And another thing I wanted to say, there are many aspects that make working with endogenous natural molecules so beneficial. One of the things that was brought up is safety. Well, we think it's really de-risked. We shouldn't have off-target effects or safety toxicity problems. But another is that we can mine what's out there in the data about humans because they have this molecule. We can do
interactive networks. Yeah, that's very interesting.
And you know, the molecule is present in the blood, it's present in samples. So we're actually looking for somebody too that can help dig through the biobank data, UK biobank, things that are maybe publicly available, to get our hands on some of that data and start looking into that. But yeah, it's sort of it's hard to think because it is really difficult to hire somebody right now.
Yeah. Oh, well, yes, it's probably doubly difficult because bioinformaticians are already at a shortage of availability, and plus COVID making, you know, just the networking experience that much more difficult, I would imagine, right? Yeah.
But you can also, I mean, I think we're not quite at the sabermetric stage of it, but it's moving forward. So I actually did my undergrad research in silico, the Varner lab at Cornell. And we're starting to see, I don't think the promise that it's going to completely get rid of in vivo preclinical models, I don't quite buy that, but I think it can be a lot smarter and one with the 3 R's with the FDA, reduce the number of animals that you use and give you a more streamlined. But I mean, what was it Frank Doyle, who's now Dean of Engineering at Harvard? He had a big breakthrough with the modeling of the insulin pump. So I think, like Nicole was saying, especially for, sorry, Nicole, what was the phrase? Endogenous compounds?
Mm-hmm.
My brain's a little fried.
We try, you know, we try and stay away from this idea of like natural molecules, because then people think of supplements and things like that. So yeah, we say endogenous, it just sounds a bit more serious.
And yeah, and similar, obviously ours is more biomimetic than straight up endogenous, but you can also then have a better idea of what to look for, whether it's succeeding or failing, rather than you can have a microscopic view rather than just a macroscopic.
Very interesting. Actually, so I've got a couple different directions I want to go. So I'm pausing because I'm trying to figure out which direction, which thread we should follow. Let's continue down this road of how new tools will help the development, the early developmental stage process. And I wonder if you guys have thoughts around, let's say, some of these computational biology tools or artificial intelligence databases. And the reason that I ask is because of a very interesting conversation I had earlier this week with a founder for a different platform that will take all the characteristics that are out there for a given molecule. And the different databases, they mine also the literature, they mine the different clinical endpoints to look at what has been done with similar molecules. And all of this is to build a probability of success for molecules as they enter into the clinical stages. And I just thought, well, isn't that interesting if you have a library of molecules to know which one to put your resources into? And so I wonder if there are other types of technologies that any of you, and I'll just open it up to anyone who has thoughts here, see as moving forward. Yes, Peter, I see you raised your hand at that.
Sure. So I mentioned before that I was responsible for Merck's external medchem. And as part of the exercise of doing this, I was actually tasked with, if we wanted to create an external group that was independent of Merck, but owned by Merck, what will we go and do? And the long and short was, the one thing that we felt we couldn't outsource at the time was the starting point, the sample collection. Now, mind you, this was 10, 12 years ago, so IT is not where it is today. But that exercise taught me that the starting point that you pick has a profound impact on where you end up. And Nicole pointed that out very nicely with the endogenous compounds. That's exactly the point she's making in my perspective. So we're constantly looking at tools to improve our starting point. And this is actually one of the reasons we were able to negotiate the license with Schrodinger. Schrodinger uses us as a beta testing library. So we get access to things that are cutting edge to use in our community, because they can use that to refine it to help others that are commercial partners. These include AI screening libraries, these vast computational libraries against stocking. And when we do these things, we tend to do them against what we would view as traditionally non-competitive targets. So we're actually doing one on TB. So Schrodinger has never actually done one of these types of massive libraries where you're literally computationally docking billions of molecules. But we pick one because it would limit our, if we pick a commercial target, if you will, then there's issues of publication and patenting. But if you go and pick a public health need and we work on these, then we can very quickly move from the screen to the molecule with the whole idea of actually doing exactly the virtuous things you're talking about. Better starter point, faster place to an animal, higher probability of success. I have limited ability to do a lot of the AI things that you were talking about, and I would love to do it. I don't have the resources, but something like this would be a huge game changer. And it includes screening against the audience at large may or may not be aware that one approach to virtual screening is for chemists to design libraries of theoretical molecules that they could make quickly, but have never made, but would be vast. And then use the computational, that's right, the computational tools against a never made set of molecules, which is hundreds of millions, if not low billions. And in the event of a hit, in a month or 2 or 3, not very long, you could actually make that library because your confidence in the chemistry. This is part of that work. So that's the closest I get to it. I view it as really valuable that the cost of actually paying for the actual chemistry and biology side, I approved off of a 1-hour phone conversation, because I see that if that works, this would be worth hundreds of thousands of dollars to me for my future programs that I would happily pay.
Right, right. You can certainly see the potential in something like that. Absolutely.
Plus, it's
Nicole.
It's TV.
Yeah, I mean, going back to the issue of trying to decrease the number of animals that we use and decrease the huge costs of these preclinical studies. I mean, this is innovation that's really important for us. And you know, this is beyond in silico now. Like this is, you know, really, we're talking numbers of animals, and it's so difficult in some fields. I mean, neuroscience, we are so far, it feels like we're so far behind because we don't even know what causes these diseases. And then our mouse models for them are not even, they're not really even real models. Mice don't have Parkinson's disease, and yet we have to spend hundreds of thousands of dollars and use many mice to try and show in vivo proof of concept. And we are doing that right now. I mean, these studies are really, really long, and you know, and pharma, they even use like non-human primates. I mean, this is crazy, really.
Right, right. Especially for these early stage candidates when you're not sure whether it will be worthwhile to move forward because of something like toxicity or something, you know, not even necessarily for efficacy.
Yeah. And so, you know, this is why we as a company, we're pushing forward these in vivo proof of concept. They're nice to have, but they're not pivotal. We are pushing forward. We have confidence that our molecules are not going to be toxic, but yeah, we still have to do the two different animal IND enabling studies, and that I don't think it's going to change. Although there's a real will for that. One thing that I do think is going to help a lot are things like using stem cells, using organoids instead of animals. But it still feels, especially in CNS and neuro, it feels very, very far away.
I would imagine.
But yeah.
Yeah, no, and I'm clapping my hands here because Nicole, you set me up so nicely yet again for the next question that I had, which was what tools do you see on the horizon? And I'd kind of like to hear from all of you. It sounds like we have a very various expertise here, but I was thinking in my mind along the lines of organoids that would help us to screen, you know, without using those animal models, at least in the very early stages. And I wonder, I think that with 5 minutes left, I'd like to kind of leave the audience with a sense of what is coming and some of those technologies that are on the horizon to help us in the discovery and early stages of development processes. And I could make a couple more suggestions of things that come to top of my mind, but I really want to hear what you guys have to say. And again, I'll step back and see if anyone has thoughts without necessarily calling on folks right off the bat.
Sure, I'll just chime in very briefly. I believe strongly enough in organoids that when Weill Cornell built an organoid platform, that actually my group was the first user of it. We recognize that it's the same problem that Nicole is talking about, that these are likely to be more translational than the animal models. And so I'm willing to fund my part of this core to have access to the technology, because we need to eke out every bit of efficiency we can. And if we can move the needle even a little bit of our success rate, it will have an incredible societal impact.
And could you just add a little more detail, Pete, to why you think those organoids are going to be more translational than some of these animal models?
Well, it turns out that for the scientists that actually go and study the organoids, the 3D nature of it, they actually start building much more of the matrix that actually exists in an actual tumor for the sake of argument, but it doesn't have to just be a tumor. It can be used for other disease states. And so when we've traditionally looked at a cellular system, so a confluent monolayer of some sort, you're hitting your target, but the behavior in an organoid could be profoundly different. The translation of that, just sticking to cancer because it's the easier one and it's where we've been working, but it's not limited to that. I don't know how many times my teams at Merck and at TDI have shown regression of a tumor model in a mouse and have it be ineffective, and that's the right word, in a clinical study. And so it's actually been my understanding that places like Amgen, actually, if they have a choice between an animal model or an organoid, if they can only do one, they're going to do the organoid because they believe it is more likely to predict the efficacy that you ultimately see. You are recapitulating much more of the structure that exists in patients, is ultimately what it comes out to.
Yeah, very, very interesting. Now, Nicole, do you echo the organoid excitement, or where do you really?
Yeah. And there are exciting new companies that are working on this, and yeah, a good number of companies. Another thing that we really need technology around, and have needed for a while, are diagnostics, right? And it's not as sexy, it's not as trendy, right? But we still need diagnostics, and we need there to be investments in diagnostics. For us, we have a neuroprotective molecule. We want to actually delay the onset of the disease and slow the progression of the disease. But at this point, there's no way to clinically test delay of onset and neuroprotection, because we can't identify the patients before they're symptomatic.
Right.
And
right.
So this is a huge problem. And again, there are promising, interesting companies working on it, but they do sometimes come up against a wall. It's investment. It's they struggle too.
Yeah, absolutely. And thank you for bringing up that important aspect of not only being able to treat something, but detecting it and understanding what the root cause is certainly an underserved problem. Damien, thoughts on what tools you're excited to explore moving forward as you expand the Cayuga Biotech portfolio and move forward with your lead candidate molecule?
So obviously, I'm biased, but in general, I think nanotechnology is going to be the big leap forward. Peter was already talking about he has compounds that work, but they're too toxic. So nanoparticles, be it cancer, be it hematology, I always joke that we're about 20 years behind what you can do with cancer drugs, but it's really how you can get it delivered. You can have it degrade over a longer time, so you have a longer term of effectiveness. I think it's not going to solve everything, but it can make things a lot better. And in diagnostics, so you've got the Cornell dot from Elucid Oncology for brain tumors. There's just it can, if it's done right, it can help in a lot of different fields. So it's very versatile.
Wonderful. Well, I would like to say thank you to all of you for leaving us with a little bit of hope for technologies of the future and for sharing your thoughts around where our future drugs are going to come from. So thank you very much, Nicole, Pete, Damien. This has been a lot of fun for me, and I hope that you also enjoyed the conversation.
Yeah, thanks for all your hard work pulling this together. This was fun. And yeah, thank you.
Great. Hopefully we can all meet again in, together over, a frosty glass of like happy hour beer or something like that in the future, which is where we hope these fundamental questions will be moving in the,
No one would object to that.
Yeah. All right. I'd also like to say thank you to Alex Hall-Daniels and Josh Torrey, because they've been running AV for these virtual events. And thank you to our audience for participating. We will be back next week. We'll be talking about commercialization around technology in public health applications. So that's very exciting. And again, thank you to our panelists, and have a good night, everyone.
Good night.
Sure. Thank you so much.
Take care.
Right.
Take care.